Good morning, everyone. Thank you so much for joining us this morning. I'm Alex Howard. I'm the Publishing Operations Manager for the Microbiology Society. I'm also the project manager for this really exciting project. So I wanted to start with introducing our sound science and open access journal, Access Microbiology. We launched this journal in 2018 to offer researchers an opportunity to publish research that is often lost due to not being seen as high impact. This means that the journal publishes original research across the full spectrum of microbiology, but it also publishes replication studies, negative or null results, and addition to established methods. Publishing this type of research prevents research waste, where experiments are redone in multiple labs for no gain. The publication criteria is therefore based on methodological rigor rather than novelty or impact. It is this journal that we're going to convert to an open research platform. Well, what is an open research platform? We describe it as a publishing platform that delivers peer review transparency and a fast track for communicating research, maximising the potential for impact and influence. And I'm going to walk you through exactly what this means in practice in a moment. Importantly, why are we launching an open research platform? Well, as we've seen over the past year during the pandemic, there has been a complete overhaul in the way research is being both undertaken and shared. Researchers are needing to rapidly share their work, often before publication, and are increasingly being required to share the underlying results to their research too. This has put the open science agenda at the forefront for the Microbiology Society, and we recognise the need for a trusted place for researchers to disseminate their work rapidly, rigorously, and transparently. In the summer of 2020, we won funding from the Wellcome Trust and Howard Hughes Medical Institute to address these challenges. The sound science and broad scope of Access Microbiology makes it the perfect journal to convert into an open research platform. To be able to highlight the benefits of our open research platform, I want to briefly go over what most journals currently offer. Firstly, but not always, authors post their manuscript to a preprint server, such as bioRxiv or medRxiv. Then, separately, they submit to a journal and it undergoes full peer review. Most of this process is hidden from the author and the public. It is only right at the very end of the peer review process, when the article has been formally accepted, does a final version of the article get published in the journal. What this means is that very little is known about the entirety of the peer review process, and the preprint and published article are in completely different locations, which means that any citations accrued on the preprint on bioRxiv, for example, are separate from those citations on the published article in the journal, essentially diluting an author's citations. So what makes Access Microbiology's open research platform so special? Well, as soon as authors begin the submission process to Access Microbiology, they can receive feedback from a series of artificial intelligence or machine learning review tools. For example, one of the tools we're considering can be used prior to submission to ensure the article complies with the platform's ethical guidelines and policies. Running an article through this means that authors are much less likely to miss key requirements and have their article returned to them at the very first hurdle. Then, during the submission process, another tool scans the methods for a variety of rigor criteria which have been shown to contribute to the reproducibility of research. It also detects things like sequencing data, plasmids, cell lines, and software tools, and assesses how uniquely identifiable and accessible these are. These are things authors can easily include but have not been trained in the importance of, and as such their work is less reproducible. A third tool scans the article for language, picking up on text that may have been unknowingly reused from previously published work. Our testing of this tool identified that a surprising number of authors do this, and it is something that is easily rectifiable. All authors can choose to see the reports from these tools and immediately revise their article before it is posted on the platform. We really see these tools as providing a unique service to authors in helping them improve their article right from the very beginning of the submission process. Once the author and editorial office are happy with the article, it is immediately posted onto the platform as a preprint, as well as the AI review reports. The preprint can start accruing citations and others can provide feedback on the work. Plus, making these AI review reports available alongside each preprint increases transparency and may help readers assess the validity and reliability of the preprint themselves prior to full peer review. Meanwhile, the article goes through a full, transparent peer review process cycle. On Access Microbiology's open research platform, the editor's decisions, reviewers' comments, preprint versions, and author responses are all posted publicly, increasing transparency and promoting trust in the peer review process. When the editor considers the article to be of sufficient scientific rigour and quality to be accepted, this is then fully typeset, copy edited and published as the formal version of record, or final article. It is then indexed in PubMed Central and other indexing services. This version of record is linked to all previous versions, meaning citations are accrued. So our open research platform offers a single platform that keeps all of your citations together and provides a clear history of the peer review process. Again, what will our open research platform offer? The submission process uses our current technology and is very simple to use. Authors can choose to immediately improve their article during submission through the use of the AI review tools. Research can be rapidly disseminated as a preprint. Having transparent peer review promotes trust in the review process, whilst reviewers can also get public recognition from their reviews. The process is unified, meaning your versions are in one location and citations are accrued. The platform will publish all microbiology research as long as the science is sound. The open data policy means people can easily reuse and cite your work, increasing reproducibility. Publishing with our platform directly supports the microbiology community by funding Microbiology Society grants, outreach, and policy work. What would I really like you to take away from this webinar? Well, the more open the science is, the more rigorous and reproducible it is. We will be launching the platform October 2021. It will be completely free for authors to submit and publish for the first 12 months. This is because the Wellcome grant is funding the first year of running costs, so we can subsidise all the cost for authors. And finally, this open research platform is being developed for you, the microbiology community. The more feedback we have, the better it'll be. So please do get in touch to let us know your thoughts. And now I would like to hand over to some of our members who would like to share their thoughts with you on our open research platform.
I think pre-printing is a fantastic idea to get science out there so that we're no longer competing, we're taking advantage of each other's data and helping each other and facilitating the research going forward rather than just being in a competitive and a what have you done, what have they done sort of environment.
I'm very much in favour of this model because it's fundamentally subversive. One of the hardest barriers to achieving diversity, equality and inclusion within societies is conventional hierarchies of authority, and this is as true in biological sciences as it is in society as a whole. So we need much more democracy and opportunity of expression. While Twitter has provided us with an element of this, we really need science-oriented platforms that encourage the judgment of quality and significance being returned to the reader back from editorial opinion polls. To that extent, the open research platform is both disruptive and egalitarian in a way that I think can be really constructive for original and innovative science.
So I just want to spend a minute just explaining how I see kind of us in the microbiome research that we do and other microbiome researchers potentially using the open research platform by the Microbiology Society. So we do a lot of clinical studies, and in fact in the clinical kind of arena it's really important to also show negative results. So I can see the open research platform being an ideal place to actually disseminate the information from these studies. And I think the other thing that's really important to say is we also need to share kind of negative or results that confirm previous findings. And again, this is really important in the context of microbiome research to kind of make sure we're not overhyping different kind of associations, but also to make sure what we're seeing is a really strong association so we can potentially move forward into the clinic. So again, I think the Microbiology Society with this new kind of open platform is the ideal place to kind of deposit these findings and this data so it can be widely accessed by the wider research community and kind of really make positive strides in the microbiome research space.
If you would like to know more about the project, I will be in the exhibitor hall as soon as this webinar finishes, and I will be there until 3 p.m. this afternoon. So please do come and chat to me. I would love to hear what you think and answer any of your questions. With that, I would really like to thank you for joining us this morning. Please do follow the Microbiology Society Twitter account. We will be doing regular monthly blogs on the project, and we'd love to get you involved and keep you up to date as much as possible. Thank you again.